Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a future where the fear of hidden gluten, that constant checking of food labels, just isn't a daily worry for millions.
Yeah,
We're diving into some really exciting new research today. It offers a real glimmer of hope for a treatment for celiac disease.
exactly. A condition that currently means a pretty restrictive life for so many.
Right.
And this deep dive, it really takes us into a potentially, groundbreaking area in autoimmune research, celiac disease, where the immune system reacts badly to gluten.
That protein in wheat, barley, rye.
Precisely. It affects a huge number of people globally, maybe around 70 million. And right now, the only management is that strict, lifelong gluten-free diet.
Which, as you probably know, can be really tough.
Yeah.
Social isolation, nutritional worries.
Sometimes symptoms still pop up, even when you're being super careful. It's frustrating.
Definitely. Okay, so let's get into the specifics of this potential game changer. We've looked at a couple of things. First, a health news article, pretty accessible, reporting on a new study. And then the scientific paper itself, the peer-reviewed one.
Right, the source material.
Which lays out all the detailed experiments. Researchers in Switzerland did this work.
And their focus is on this really interesting idea, sort of a cell-soothing technique, targeting those regulatory T cells, or Tregs as they're often called.
So for you, the learner, our goal here is to really unpack this research.
Yeah, we want you to understand what it could mean for people with celiac disease, what hurdles are still there, and why this is actually quite exciting right now.
Okay, straight to the point then. What was the main aim? What were these scientists actually trying to figure out?
Well, the core purpose was to see if these engineered regulatory T cells could be a viable therapy for celiac disease.
So using modified immune cells.
Exactly. Could they use these cells to sort of retrain the immune system to tolerate gluten, to offer a real alternative to just avoiding gluten for your whole life?
Okay, and how did they go about testing that? What did the experiments look like?
They took a two-pronged approach, basically.
Sure.
First, they used an animal model.
Mice.
Yep, mice, specifically bred, to have a genetic setup similar to humans with celiac disease. They carried this human gene, HLA-DQ2.5, which is strongly linked to celiac.
Ah, okay.
Then, the team created two kinds of T cells in the lab. Effector T cells, those are the ones causing the trouble, the inflammation.
The attackers, sort of.
Right. And regulatory T cells, the ones that normally keep the effector cells in check.
And they engineered these.
They did. They modified the receptors on these cells so they specifically recognize certain bits of gluten epitopes, they're called. They zeroed in on two key fragments known to cause a strong reaction in most people with the condition.
So they programmed immune cells to react specifically to gluten components. What happened when they put them in the mice?
Well, they used a technique related to something called chimeric antigen receptor T cell therapy. You might have heard of it in cancer treatment.
Yeah, where they engineer immune cells to fight cancer.
Exactly. Same basic principle here. Modifying immune cells, but aiming for tolerance to gluten, not attacking cancer.
Got it.
So these engineered T cells were infused into the mice. Then, the mice were given gluten, specifically a known reactive peptide from gliadin.
And then they watched what happened.
They tracked everything very carefully. Where the engineered cells went, if they multiplied, how they interacted in the gut and lymph nodes. Crucially, they looked at whether the engineered regulatory T cells could dampen the response of the engineered effector T cells when gluten was present.
Sounds incredibly detailed. You mentioned a second part, with human cells.
Yes, that's right. They also worked with human cells directly in the lab. They isolated different human T cells, including regulatory T cells, from blood.
And engineered those too.
Just like with the mouse cells, yeah. Modified the receptors to be gluten-specific. Then they did in vitro tests, think test tubes, petri dishes.
Lab bench stuff.
Exactly. To check if these engineered human cells worked as expected.
Right.
If they could actually suppress other immune cells in the presence of gluten. They even compared their performance to the natural regulatory T cells we all have.
Okay, so a lot of careful work, both in animals and with human cells. What were the main discoveries, the key findings?
The results in the mice were quite encouraging, actually. They saw the engineered effector T cells, the attacker cells, did indeed travel to the intestines and multiply when the mice got gluten.
As expected, perhaps.
Right. But when the engineered regulatory T cells were also there, they essentially put the brakes on. They stopped the effector cells from migrating as much and reduced their reaction to the gluten.
That sounds promising.
And here's something really interesting. Regulatory T cells designed for just one specific bit of gluten could also suppress effector T cells reacting to a different bit of gluten.
Oh.
They call that bystander suppression.
That bystander effect seems pretty important, especially since gluten is so complex, right? Lots of different parts that can cause a reaction.
Exactly. It suggests a therapy targeting maybe just a few key parts of gluten might still offer broader protection.
What about the human cell experiments in the lab? What did they show?
Well, in those tests, the engineered human regulatory T cells were actually better suppressors than the natural polyclonal regulatory T cells, at least when activated by that specific gluten piece they were designed for.
Better than the body's own regulator.
In that specific context, yes. They also found a link. The better the engineered receptor was at grabbing onto gluten its functional activity, the better the regulatory T cell was at suppressing the immune response.
Makes sense.
And finally, the study also mapped out some specific interactions happening between the gluten-specific effector T cells and other immune cells called antigen-presenting cells, specifically dendritic cells and B cells, both in the gut and the lymph nodes.
Okay, this is where it gets really exciting. Let's talk implications. What could all this mean down the line for people living with celiac disease?
Well, the biggest potential implication, clearly, is a whole new treatment approach.
Right.
The success in the animal model, it really points towards cell therapy using these engineered Tregs.
Moving beyond just diet.
Potentially, yes. Moving beyond relying solely on that strict gluten-free diet. If this pans out, it could drastically improve quality of life for millions. Freedom from the constant dietary vigilance, the social impacts, the health worries.
Absolutely. I mean, just imagine eating normally without that fear. Apart from a potential treatment, what else does this research tell us?
It also gives us some really valuable clues about the basic mechanics, the steps involved when the immune system gets triggered by gluten in the first place.
Understanding the process better.
Right. And that bystander suppression finding we mentioned, that's really significant. Gluten isn't simple. It's a mix of proteins.
Yeah.
The idea that Tregs targeting just a few key bits could calm the whole response is, well, very hopeful. It suggests you might not need to target every single reactive part of gluten.
Like going after the ringleaders might quiet down the whole gang.
Sort of, yeah. It's a promising angle for therapy design.
And you touched on the link to cancer therapy techniques earlier. How significant is that connection?
I think it's quite telling. Adapting methods from chimeric antigen receptor T cell therapy, that cutting-edge cancer immunotherapy, shows how breakthroughs in one medical field can spark innovation in totally different areas, like autoimmune diseases.
Cross-pollination of ideas.
Exactly. It highlights how interconnected medical research can be.
Okay, this sounds incredibly positive, but research always has limitations, right? What are the caveats here? What should we keep in mind?
Absolutely. It's crucial to be realistic. First off, the animal model. While useful, it wasn't a perfect replica of human celiac disease.
How so?
These mice didn't automatically get the intestinal damage seen in humans just from eating gluten. They needed the engineered effector cells to kickstart that process. So we need more research in models that better reflect the active disease state in people.
Okay, that's a key difference. What else?
The experiments in the mice were also quite short-term, just 5 days. We don't know yet about long-term effectiveness.
Right.
Will these engineered regulatory T cells stick around? Will they remain stable and keep working over months or years? There is a potential concern they might lose their suppressive power, especially with repeated gluten exposure. That needs investigation.
And the timing. Did they look at when to give the therapy?
No, the study didn't explore the optimal timing. In some other autoimmune models, giving Tregs before an immune response kicks off seems to work differently. So that's another open question.
And what about safety? We're talking about putting engineered cells into people eventually.
Safety is paramount, of course. They use specific viruses, adeno-associated viruses, to get the genetic instructions into the T cells.
Is that standard?
It's a common tool in gene therapy research, generally considered relatively safe. However, the long-term safety of this particular gene editing method in humans, especially potential unintended genetic changes, hasn't been fully assessed for this application.
So hurdles remain before human trials.
Definitely. This specific approach using these viruses isn't quite ready for prime time in humans yet. More safety work is needed.
One more thing, celiac genetics aren't uniform, are they? Did the study address that?
That's a very important point. The study focused heavily on that HLA-DQ2.5 gene variant.
Which most people with celiac have.
Over 90%, yes. But there are other HLA types linked to celiac, like DQ2.2 and DQ8. A future treatment might need tailoring for people with those different genetic backgrounds to work for everyone.
Right, that HLA-DQ 2.5 creates a specific sort of docking site on immune cells, making them good at presenting gluten fragments to those inflammatory T cells.
Exactly. So they focused on that major player, but the others matter too.
It's vital to get the full context. So, what did the actual researchers say about their own work and the next steps?
Dr. Raphael Porret, the lead author, was pretty clear. He said the study gives proof of concept evidence that engineered Tregs could potentially restore gluten tolerance.
Proof of concept, so early stage but promising.
Yes. But he also stressed that much more work is needed, especially looking at how well it works when the disease is already active and causing damage.
Makes sense.
He also mentioned the study helps us better understand those initial steps in how the immune system gets activated by gluten after you eat it.
So, a cautious optimism but acknowledging the road ahead. Anything else from their discussion in the paper?
They did point out that the whole role of regulatory T cells and how celiac disease naturally develops is, well, still a bit murky.
Still debated.
Yeah, somewhat controversial, or at least not fully understood. They also reinforced how crucial it is for these engineered Tregs to be highly specific to gluten to be effective. That antigen specificity is key.
And what about the natural Tregs?
The paper touches on observations that naturally occurring gluten-specific Tregs might be rare, or maybe don't function quite right in people with celiac disease. It's complex.
I see.
The researchers proposed that maybe blocking the chatter between T cells and B cells, another type of immune cell using these engineered Tregs, could be a core strategy.
Interesting angle.
And finally, they noted that how you design the gluten fragment used to activate the engineered receptor really matters. Higher activity seemed to correlate with better suppression, echoing findings in other autoimmune research fields.
This has been a really insightful deep dive. It's clear there are still challenges, absolutely, but the potential upside here is just massive for so many individuals.
It really is. This research offers a genuinely exciting peek into a possible future where managing celiac isn't just about avoidance.
On that note, here's a final thought for you, the listener. This work obviously sparks questions about the future of treating autoimmune conditions. What strikes you as the most critical next research step to get therapies like this from the lab into clinics? Maybe you're curious to dig deeper into how that chimeric antigen receptor T-cell therapy actually works, or perhaps explore the different flavors of regulatory T-cells and their jobs in the immune system. It's all part of that bigger journey to understand and treat these complex diseases.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.